I'm working as scientist in ICAR-CTCRI. CTCRI is solely working on tropical tuber crops. So you have heard of tropical tuber crops like cassava, sweet potato, yams, and tubers. And these are known generally for its storage content, that is carbohydrates. But in our germplasm collection, we have a vast number of aromatic and colored tubers, which can provide additional nutrition in addition to the non-carbohydrate content. So that is why I am interested in this topic, Nutrigenomics. So I'll be covering the topic, what is Nutrigenomics, the principles behind Nutrigenomics, nutrients, and some examples of its health implications, mechanism of nutrient-diet interaction, tools to study nutrigenomic interactions, limitations and scope of nutrigenomics, and the goals of nutrigenomic study. The concept of food as decisive factor for health is well established from ancient times. We have all heard the saying, let food be thy medicine and medicine be thy food. This is attributed to Hippocrates. And we have sometimes seen cats eating grass. Plant eating is a normal cat behavior that provides cats with dietary fiber to act as a laxative. This shows that inherent knowledge that diet can control disease is prevalent even among animals. Nutrition has been a central element in many traditional forms of medicine also. Today, incidence of lifestyle diseases are very high, and people has always had and still have an urge to control lifestyle diseases through diet. The concept that diet and health are related is now well understood. And what has evolved in the concept of food as medicine is the level of understanding of how food regulates health, and this comes from the advances in the NGS technologies. Nutrigenomics attempts to study the genome-wide effects of nutrition. Or in other words, nutrigenomics helps to understand the language that diet uses to message our genes. If you understand this language, you can regulate how food interacts with your body and optimize your health. Definition of nutrigenomics goes like this. It is the prospective analysis of differences among the nutrients in the regulation of gene expression. Consequent to gene expression, it also regulates mRNA synthesis, protein synthesis, and metabolite production. The excitement about nutrigenomics comes from a growing awareness of the potential for modifications of food or diet to support health and reduce the risk of diet-related diseases. So there is some basic principles for the nutrigenomic study. That is, the nutrients in the food can directly or indirectly affect the genome through change in its structure and expression. Diet can occasionally be a risk factor for developing disease in some individuals. Some genes, under the regulation of bioactive components in the diet, play an important role in onset, incidence, and progression of disease. The extent to which diet may affect balance between health and disease depends on the individual's genetic makeup. Nutritional intervention is dependent on nutritional standards and needs of the individual as well as individual's genetic makeup. An awareness of nutritional genomics can ideally prevent, mitigate, or heal chronic diseases. Nutrigenomics focuses on how nutrients or non-nutritive bioactive components such as enzyme inhibitors found in the diet affect gene expression, protein and metabolic concentration, and therefore metabolism, health status, and risk of disease. This is a part of nutritional genomics that incorporates nutrigenetics as well. Nutrigenetics is the analysis of genetic variation among individuals with respect to interaction between diet and disease. That is, nutritional genomics incorporates two subjects. Nutrigenomics that analyzes the effect of nutrients in the genome, effect of nutrients in the gene regulation, and nutrigenetics that analyzes the genetic variation among individuals. The case of lactose intolerance is an example of nutrigenetics. Nutrients primarily enable normal development and functioning, and pharmaceuticals are generally developed to treat, cure, or prevent disease. Now, nutraceuticals are coming, and these are also considered as nutrients, and they can have health implications. So, in the term nutrigenomics, one term is nutrients and the other is its effects on genome. So, first we will see the nutrients. Nutrients includes macronutrients that make up the bulk of foodstuff that has primary role in providing energy. That includes carbohydrates, fats, and proteins. And this also has role in nutrition also. Although it provides energy, it signals many other pathways. Micronutrients that consist of organic and inorganic compounds present in small quantities, not used for energy, but needed for good health. These includes essential vitamins and minerals. And also the third component of nutrients is secondary metabolites, which is the metabolic products of macro and micronutrients. Unlike ubiquitous vitamins and minerals, these are specific and unique to certain plant species or genera, but they have immense health properties. Sometimes the properties are anti-nutritional and allergic, and sometimes it is very much beneficial like antioxidant effects. Many phytochemicals have. So I was saying that we have to see the two components of that word nutrigenomics, and it includes nutrients and genomics. And nutrients includes macronutrients and micronutrients. Macronutrients make up the bulk of foodstuff that has primary role in providing energy. That includes carbohydrates, fats, and proteins. And micronutrients includes organic and inorganic compounds present in small quantities, not used for energy, but needed for good health. These includes essential minerals and vitamins. And coming to the other part is metabolites of these macro and micronutrients, that is secondary metabolites. Unlike ubiquitous vitamins and minerals, these are specific and unique to certain plant species or genera. This is what I said. We have in our tubercles germplasm many tubers with specific content of secondary metabolites that is indicated in their phenotype as aromatic compounds and colored features. Many phytochemicals have significant role in human health. A large population in developing countries has as their staple food cassava, wheat, rice, and corn. These are poor sources of some macro and many essential micronutrients. Here is where we can intervene and enrich our crops like cassava, wheat, rice, etc. with more nutrients. That is where one application of nutrigenomics comes into play. Even in developed countries where food is abundant and daily calorie intake is high, the poor eating habits of people lead to micronutrient deficiency diseases. All these factors again point to the factor that nutrients regulate the state of well-being. The patterns of gene expression or protein expression or metabolites in response to particular nutrients at a given time in a particular cell or tissue is regarded as dietary signatures. Such dietary signatures act as biomarkers which can be used for diagnosing at an early stage the onset of some diet-related disorders. So we now know what is nutrients, and we have to see how these nutrients are acting on the genome. Initially, it was thought as mainly a factor of metabolism. It's a chemical entity that is involved in the metabolism. A nutrient is not merely a chemical component required for a particular metabolic function. It also plays an important informational signal or signaling role in the cell. As with any system that transmits information, the signal must have a sensor or receiver that can accept, decode, and relay information that has been transmitted. Cellular proteins that receive and transmit this information are termed as receptors. So when a nutrient comes, it acts as a signal, and that signal is received by the cellular proteins. The receptors then must relay this information via a transducing mechanism to the part of cell that is capable of reprogramming the cell to adapt to the new environmental conditions. The signals received by the cell will be transferred to either to cytoplasm or nucleus where its action will happen. This reprogramming can occur in the cell nucleus or cytoplasm. It can involve changes in the expression of genes through transcription and translation, and it can affect the stability of messenger RNA and protein, or the activity of proteins will be affected. So mode of action of nutrients can be summarized like this. Nutrients interact with receptors, activate or modulate transcription of target genes, or directly cause the rearrangement of chromatin structure. These nutrients play a role in metabolism and also has role in signaling. It regulates transcription, translation, and epigenesis. The action of nutrients over genome can be direct interactions. Nutrients sometimes, after interacting with the receptor, behave as a transcription factors that can bind to the DNA and actually acutely induce gene expression, or it can repress the gene expression. Epigenetic interactions. Nutrients can alter the structure of DNA or of histone proteins in chromatin so that gene expression is chronically altered. Genetic variations. Common genetic variations like single nucleotide polymorphisms can alter the expression and functionality of genes. To reiterate, nutrients act as a transcription factors, hinder or promote gene expression. It modifies the gene structure and blocks the gene expression. It affects the concentration of intermediaries of metabolism and substrates that can act as cofactors promoting gene expression. It can alter signal transduction pathway. Now we will see some examples of dietary nutrients in gene regulation. First, carbohydrates, we all know, is one of can be glucose monomers like glucose or other polymers, and dietary carbohydrates has role in obesity, diabetes, and these two are common lifestyle diseases. Until recently, carbohydrates was regarded as chemical entity that alters the metabolic pathway. It is now inferred that it plays a crucial role in cell signaling, which controls the level of insulin. In pancreas, glucose is primary stimulus regulating insulin synthesis and secretion. In liver, in presence of insulin, glucose induces expression of genes encoding glucose transporters and glycolytic and lipogenic enzymes. And it represses genes encoding enzymes in gluconeogenetic pathway. This is how the glucose level is maintained. Vitamins. These are another group of micronutrients. They function as coenzymes, cellular antioxidants, and regulates gene expression. Vitamin deficiencies or overintake affects carbohydrate, nucleic acid, protein, fat, or mineral metabolism. The deficiency of many vitamins mimic a condition of radiation damage to DNA, causing single or double-stranded DNA breakage or oxidative damage or both. So some of the nutrigenomic effects of vitamins and minerals, for example, deficiency of vitamin B2, B12, folic acid, B6, niacin, C, vitamin C or vitamin E or iron or zinc appears to mimic radiation damaging DNA, and it is caused by single or double strand breaks or oxidative lesions or both. Folate deficiency breaks chromosomes due to the substantial incorporation of uracil in human DNA. Single strand breaks in DNA are subsequently formed during base excision repair with two nearby single strand breaks on opposite DNA strands, leading to chromosome fragmentation. Folate is a nutrient needed by the body to make DNA. When you do not make enough folate, you have a higher risk of developing cancer. So, if through nutrigenomic studies we are able to understand the mechanism of action of folate, and if through nutrigenetic studies we understand that particular genotype is having a lack of folate, we can supplement folate in this diet, his or her diet, and make him stable. Some of the examples of nutrient deficiency and the gene alteration that causes is like this. Folic acid and vitamin B12 causes chromosome breakage and DNA and affects the DNA repair methylation. Niacin hampers DNA repair. Vitamin E mimics radiation damage. Zinc affects the chromosome and causes breakage in it. Fatty acids alters gene expression. Secondary metabolites also has an indication in regulating genes. Some of the examples are carotenoids. They have a role in reducing cancer, age-related macular degeneration by antioxidant mechanism, preventing free radical generation. Glucosinolates target drug metabolizing enzymes and leading to decreased carcinogen DNA interaction and increased detoxification. Phytoestrogens are like isoflavones, lower the osteoporosis and coronary heart disease. Another example is resveratrol. It is commonly contained in red wine, and it is a nutrient that stimulates gene that protects the tissues from free radical damage. Now we have seen what is nutrients, how the mechanism of action of nutrients is, and now we have to understand. For understanding the mechanism of action, we have to use some tools. The basic principle in understanding these tools involved in understanding this mechanism of action is central dogma, that is DNA through replication generates DNA, and through transcription generates RNA, and RNA in turn through translation generates protein. We may use molecular biology for identifying the conditions that lead to the amplification of genetic information and synthesis of several copies of RNA, and ultimately the initiation and progression of protein synthesis and metabolite concentration. The hierarchy of molecular control of biological systems passes from gene through gene modification, that is epigenetics, to gene expression, that is transcriptomics, to protein expression, that is proteomics, to metabolites, and that is metabolomics. Finally, we will be seeing the effect as phenotype or outcome. The new tools in the post-genomic era opens a new future for nutritionists to screen the background, monitor the transcriptome, proteome, and metabolome, and develop dietary strategies which are targeted to supply optimum nutrition for single individuals. So some of the tools are like this. For genotyping, genome-wide analysis through NGS technologies that can that can detect a single nucleotide polymorphism through microarray can be used. Transcriptomic effects can be studied through microarray techniques and RNA-seq. Proteomic studies can be studied through SDS page, 2D gel electrophoresis, TIGE, NMR, mass spectroscopy, and metabolic effects are also studied through high throughput methods like NMR and mass spectroscopy. Measuring the gene expression. DNA microarray technology and quantitative real-time PCR have successfully evaluated interaction between diet and gene, measured as changes in gene expression. Profiling mRNA transcripts can give nutrition scientists a holistic gene expression status in response to dietary components. DNA microarray uses hybridization-based method and RNA-seq uses NGS-based technologies. As I said, DNA microarray uses hybridization-based technology. It hybridizes cDNA with a spotted oligonucleotide probes. DNA microarray can be used for analysis of gene expression and genotyping for point mutations, SNPs, and short tandem repeats. Knowledge of genomic sequences is a prerequisite and therefore it cannot be applied widely for unknown sequences. High abundance of certain transcripts can cause noise due to non-specific hybridization. So bulk of data generated cannot be clearly used in case of DNA microarrays. This problem is overcome in RNA-seq technology, which is an NGS-based technology. It can simultaneously identify numerous gene expression in response to specific diet or nutrients. The steps in RNA-seq involves isolating good quality RNA from the targeted sample, harvesting the mRNA, construction of cDNA library, sequencing, processing the raw data to get high quality reads that may be mapped against the reference genome if it is available, or otherwise it can be used for de novo assembly of transcriptome. The data on differentially expressed genes obtained through softwares can be further interpreted. The proteomic studies involves high throughput proteomic techniques like mass spectroscopy and NMR. For small quantity protein analysis, we can use the previous technologies like 2D gel electrophoresis, SDS page, Western blotting, Edman sequencing, etc. And microarray of proteins are also available. But for understanding high throughput or bulk or entire proteome, we have to follow MS and NMR. Post-translational modifications can be distinguished by mass spectrometry. It is crucial for early diagnosis, prognosis, and to monitor disease development. Example, some blood proteins like IBP2, IBP3, etc. have been proposed as biomarkers of breast cancer, and it was detected through MS. Metabolomic profiling is most often performed using either nuclear magnetic resonance or NMR or mass spectrometry. Individual analytes within the sample are separated by their magnetic resonance shift or mass charge ratio respectively, resulting in a spectral profile of separation. MS enjoys a strong advantage over insensitivity compared to NMR via application of different combinations of chromatographic methods and mass charge separation technologies, provides the ability to measure a broader array of small molecule metabolites. Some limitations of nutrigenomics is that the subject is still in its infancy. The tools to study protein expression and metabolite production have not yet developed to the point as to enable efficient and reliable measurements. Transcriptome profiling using DNA microarrays or candidate gene analysis is a static measure of pool size of individual mRNA in a cell or tissue and does not provide any information on the dynamics of mRNA synthesis or RNA degradation. Determining the rate of mRNA synthesis typically involves Use of nuclear run-on assays that measure the relative amount of new transcripts made from previously initiated RNA polymerases and provides a measure of the in vivo rate of transcription of DNA to mRNA. Measures of RNA degradation rate are presently not easily attainable in vivo, and therefore nutritional impacts on gene expression usually are only described for the transcription rate and mRNA abundance. And the scope of nutrigenomics lies in developing of customized nutraceuticals based on specific genetic profiles. Also, it can be used for genomic-based phenotypical biomarker development that can be used to early stage detection and mitigation of diet-related diseases. So we can summarize the goals of nutrigenomics as identification of transcription factors as nutrient targets and the gene they target, identification of signaling pathways involved at cellular level and characterization of the main dietary signals, measurement of specific micronutrients and macronutrients inducing cell and organ-specific expression signatures, identification of interaction between nutrient-related regulatory pathways and pro-inflammatory stress pathways for a better understanding of diet-related diseases, identification of genotypes which can be risk factors for the development of diet-related human diseases such as diabetes, hypertension, or atherosclerosis. Use of nutritional systems biology to discover biomarkers for early detection of disease and susceptibility is another goal. So in a nutshell, nutrigenomics provides a snapshot of genes that are switched on or off at any given moment, a view of how gene-protein network collaborate to produce the observed response, the method to determine influence of nutrients on gene-protein interactions. Thank you. Excuse me, thank you.
Thank you so much, ma'am. The session was truly something all of us would have been able to learn something from. Now we shall move on to the Q&A session.
Thank you, Sarah. Ma'am, that was a great presentation. Now we have a number of doubts that have been posted by the enthusiastic participants that are waiting to be clarified by you, ma'am. Shall we move on to it, ma'am?
Oh, yes.
Okay, ma'am. Ma'am, one question that has been asked by our participant was vegans are always looking for healthy and more substantial plant substitutes to incorporate into their diet while keeping in mind the availability and budget. Is there a nutrigenomic solution to this?
Actually, the nutrigenomics provides an extended solution to this. For Jamaicans to have a nutritionally rich diet from plant source itself, what we have to do is manipulate the plant micronutrient content. For understanding what nutrient needs to be manipulated, we have to follow nutrigenomics. We will be seeing different effects of different, say for example, in tuber crops, we have 9 different types of anthocyanins. The effect of these 9 different anthocyanins will be different on different individuals and on different gene, and the role will be different as in different modes of gene expression regulation. So in that sense, which is more effective? Which anthocyanin is more effective in proper or giving the desired health effect? That we have to see using nutrigenomics. After that, we can incorporate that particular anthocyanin or enrich a particular agronomically favored crop with that particular anthocyanin. And in that sense, that nutrigenomics has a role in giving a better source of diet for this vegan people.
Okay, ma'am.
Hope I made it clear.
Yes, ma'am. Another question was posted by Sarah John, which was a lot of vitamins and minerals we know have shown anti-carcinogenic properties like selenium, vitamin C, folic acid, etc. If each of these are consumed every day at doses designated for a respective patient, will it help them stay healthy from the disease?
Nutrigenomics gives solutions for preventing diseases. If the disease has already occurred, we have to think how it will be practical to provide supplements and just reverse the effect. Studies have shown that the supplementation of these anti-cancer agents has somewhat shown some percentage of effect in reducing the onset of these cancer-related diseases, but reversing or curing effect that I have not seen anywhere.
Sublime. Ma'am, another question was, does our ancestry affect genetics in such a way that our metabolism is also affected? For example, people living in colder regions can break down fattier foods easier. Is this just a myth, or is there another explanation?
Like when people move to colder climates, that environment will have an effect on regulating their metabolism. But for when the same person returns, there is no genetic change that is happening. But if it is a person who is adapted to living in a colder climate, wherever he move out, he will have that effect. His genome is already like that, maybe because of the epigenetic factors. His genome is already like that. So may I get the question once again?
Does our ancestry affect our genetics in such a way that our metabolism is also affected?
Of course, the genetics plays a role in the metabolism, but like if you are asking for the environmental effects, that is not taken into the gene. It is only momentary. That's what I understand.
Okay, ma'am. Clear. Ma'am, most public health journals suggest that when a person suffers from cancer, mostly stomach cancer, his or her family should also be cautious about their diet and should restrict using spices. Is there a reason?
I can't comment. So how in a family, if we are following a same kind of diet, the diet will have some effect on prognosis of disease. Moreover, being a family, their genetic makeup also will be similar. So it is true that if a person in your family is having some chances of is having cancer, there are chances that you will also be having a chance of developing cancer. So this person should be cautious and he should watch his diet, but not specifically spices. I can't comment on spices.
Okay, ma'am. Another question was posted by a final year of MSc Food and Nutrition student. He or she is interested in nutrigenomics and nutrigenomics. Can you please tell him how he could do better in higher studies in this field after his post-graduation? He's interested in the field of nutrigenomics. So,
okay. As far as I know, the ICAR-CTCRI has a division of biochemistry, and they are doing good research in nutrigenomics. So if he can move to that place, and if he can get an admission there, he will be guided better.
okay, ma'am. There's another question by Shivam Pandit. Ma'am, how we are going to differentiate the effect of nutrigenomics with respect to epigenetics effect? If in case nutrigenomics doesn't change the DNA sequence, then will it be considered ever as a kind of epigenetic factor?
In epigenesis, it is not the DNA sequence that is affected, it is the DNA structure that is affected. So epigenetic factors are definitely causing nutrigenomic effects.
Okay, ma'am. Ma'am, another question. Are food restriction interventions with nutritional diets able to maintain appetite control connected to aging and defective adaptive immunity?
I didn't get the question once again, please.
Okay, ma'am. I'll repeat it. Are food restriction interventions with nutritional diets able to maintain appetite control connected to aging and defective adaptive immunity?
Are food restrictions connected to aging?
Yeah, aging and adaptive immunity, which decreases as one ages, potentially.
Definitely, when one restricts his food, his whole metabolism is altered. So it might have an effect on aging.
Okay, ma'am. Ma'am, another aspirant is asking a question related to the scope of the nutrigenomics for her future studies. So, can you explain that?
Yeah, it's an upcoming field, and I found too many researches happening all over the world. We can really contribute by for my case, what I am aspiring is what I said, like we can use nutrition, and it's a collaborative area. We can take the help of nutritional genetics, find the effect of our molecules of interest, and we can use it to manipulate our plants of interest. So it's a very of diverse scope